Get ready with me while we discuss rabies and a study regarding opossums with rabies. But first, I need to get this up. So rabies is a zoonotic disease that can spread between different species. It's caused by rabies virus, and it belongs to the family Rabies. I even said that right. It was considered 100% fatal until 2004 when Yana Geese, I probably butchered his name, was the first to survive, but it's still considered majorly fatal. Oh, if you have survived, rabies attacks the central nervous system, and those who survive are usually left with significant neurological deficits. Rabies is an enveloped RNA virus. It is rod or bullet shaped. It codes for 5 proteins. It has an unsegmented negative sense single strand RNA. You know, too late to do any of that. Oh my god. So, before signs and symptoms appear, there's an incubation period of about like 2 to 3 months, or usually less than long. But it can last from like a week to like a year, sometimes longer. Later symptoms include, like for objective, you'll see fever, difficulty swallowing, dyspnea, excessive salivation, and coma, severe paralysis. But for subjective signs, you'll see headache, nausea, hydrophobia, aerophobia, hallucinations, and insomnia. And symptoms are kind of a combination of both are agitation, anxiety, confusion, and hyperactivity. Molecular rabies enters host cells by binding to nicotinic acetylcholine receptors, NCAM, and P75 neurotrophin receptors. The virus releases its negative sense single-celled RNA genome into the cytoplasm. It uses its RNA-dependent RNA polymerase to make viral mRNA and replicate its genome. New virus particles bud from the neuron, and rabies P protein suppresses the host interferon. New virus particles bud from the neuron, and rabies P protein. The rabies P protein suppresses the host interferon signaling pathway to avoid the immune response. On a cellular level, after a bite, rabies gets into the skeletal muscle. From there, it enters the peripheral neurons through nerve endings. It travels through neurons by retrograde axonal transport to get to the spinal cord and brain, disrupting neuronal signaling and neurotransmitter function as it infects. Neurons become dysfunctional, leading to abnormal nerve impulses and neurological symptoms. On a tissue level, rabies mainly affects nervous tissue, like the peripheral nerves, the spinal cord, and brain tissue. It definitely hardcore goes after the CNS. Viral replication can cause encephalitis. Salivary glands are targeted by infection as a way to spread and infect, and it shows signs of the frothing you see. The microscopic examination can show inflammation, perivascular lymphocyte infiltration, neuronal degradation, and/or antibodies. Organ level. The brain is affected by encephalitis, which disrupts behavior, swallowing, breathing, muscle coordination, and consciousness. The spinal cord is a target for infection, and infection interferes with motor and sensory nerve transmission. Salivary glands are where the virus replicates in glandular tissue. Large amounts of virus are released in the saliva, and it spreads to the salivary glands after infecting the brain by spreading through neuronal pathways. Another thing you'll see is with the heart, it's rare, but infection can cause myocarditis or abnormal heart rhythms. On an organ system level, the nervous system is the primary target, causing later signs and symptoms. So that's when you'll start to see the telltale rabies signs. Respiratory brainstem dysfunction causes impaired breathing, muscle spasms. Muscle spasms make swallowing and breathing difficult. Respiratory failure is a common cause of death in rabies. Cat's meowing. Cat's outside the door. The immune system. Rabies avoids immune detection inside neuron. Viral proteins inhibit interferon responses. The immune system often cannot eliminate rabies before it infects the brain. Diagnosis. Definitive testing in any species requires taking a full cross-section of brainstem and cerebellum post-mortem. In animals, there's no ante-mortem tests, but approved methods. Yes, so no ante-mortem tests, but approved methods for post-mortem are direct fluorescent antibody test, direct rapid immunohistochemical test, immunohistochemistry, and real-time reverse transcription polymerase chain reactions. Humans have ante-mortem tests, but they are not sufficient for diagnosis. So samples of saliva, serum, CSF, and nuchal skin biopsies are taken. Approved methods are essentially the same things as for animals, plus rabies serology. What is direct fluorescent antibody test? It tests for rabies virus antigens in tissue. Positive rabies test shows fluorescent apple green. And direct rapid immunohistochemical test also tests for antigens through staining with anti-rabies antibodies. Immunohistochemistry looks for rabies antigens in formalin-fixed tissue using streptavidin biotin complex staining method. Real-time reverse transcription polymerase chain reaction. This test uses real-time RT-PCR methodology to detect rabies genetic material. The rabies rollout requires a cross-section of the brain stem, but skin samples from the nape of the neck and saliva samples can be ante-mortem in humans. The test targets a region of the virus genome that contains the leader region and the nucleoprotein gene. The genetic material is turned into many copies and is detected by a fluorescent probe. Rabies serology. 2 assays are used to detect rabies virus neutralizing antibodies. The rapid fluorescent focus inhibition test, RFFIT, and the fluorescent antibody virus neutralization, FAVN test. It cannot tell the difference between vaccine and infection. Neutralizing antibodies in CSF and serum from somebody who hasn't received the rabies vaccine is a positive. Treatment is mainly palliative when it is advanced. The only treatments that have cured rabies are the Milwaukee Protocol and the Racine Protocol, which is derived from Milwaukee. The Milwaukee Protocol was developed in 2004 to treat Tiana geese, the first person to survive rabies. I think she was like 15. Both protocols are similar and involve putting the patient under a medically induced coma and suppressing the brain's activity through drugs like midazolam and ketamine, and using antivirals. Ketamine also has an antiviral effect, so that was supposed to help too. Additional medications involved also include like insulin, haloperidol, amantadine, fludrocortisone, and both contraindicate the rabies vaccine and the immunoglobulin. So let's see. Putting the patient into a coma was seen as effective because it's supposedly protected from attack and saved time for the immune response, but it isn't proven to be helpful. Antivirals like amantadine work by inhibiting the production of rabies. Prevention for rabies would involve getting post-exposure prophylaxis, PEP, after being bitten. PEP is human rabies immune globulin, which is given once, and 4 rabies vaccines. The immunoglobulin is made from human donors hyperimmunized with the rabies vaccine, and they give passive immune protection until the patient produces antibodies for the vaccine. The vaccines are antibody vaccines that use a dead rabies virus for the immune system to attack and make antibodies for. There are 2 types of vaccine available. One is a human diploid cell vaccine, and the other is a purified chick embryo cell vaccine. The chick embryo, the virus is cultivated in chick embryo cells and killed with the vaccine. In diploid cell, the same is done using human cells. It's required by most states that companion animals are vaccinated as a preventative strategy. Let's talk about epidemiology, like where it's found, prevalence, stuff like that. So, rabies is found in every continent except for Antarctica and is endemic in over 150 countries and territories. Rabies causes an estimated 59,000 human deaths every year, majority in Asia and Africa. More than 95% of human rabies deaths are caused by bites from domestic dogs. And biting is the most common way to come into contact with infected saliva, so that's why you always hear about rabbit bites. In the US, 1 to 3 human rabies cases are reported annually. Because of widespread vaccination of pets, human cases are mostly from wildlife, like your bats, especially the bats, raccoons, skunks, and fox. Most US cases are from a bat or from exposure while traveling to places where canine rabies is common. Body temperature plays a factor in rabies because as opossums are less likely to succumb to rabies, they have a much lower body temperature of 94 degrees Fahrenheit compared to humans' 98 degrees Fahrenheit average. It is harder for rabies to survive at their body temperature. However, possum cases increase when there is a concentrated bat population in the area. Prognosis of rabies. I have not yet found a source that says anything other than 100% fatal, even though there have been survivors. It just shows you how few people have actually survived in comparison to those who didn't. Fewer than 40 people have survived rabies. You are likely to survive if you are treated immediately after a bite before symptoms start to show. PEP is protocol after any animal bite with an almost 100% survival rate. If you go untreated, you will die. The only thing that can lead to recovery, or the only things that can lead to recovery are the Milwaukee Protocol and the Receive Protocol derived from it. The Receive Protocol, I believe, is from Brazil. The incubation period is the period after the bite that is asymptomatic. Prodromal period is when you start to feel flu-like. Prodromal period is when the virus enters the CNS and symptoms first appear. This is when the flu-like symptoms start. Acute neurologic period is when death is near. It can be furious rabies, paralytic rabies, or atypical rabies. This is when you see biting and the telltale rabies symptoms. Naturally acquired rabies in white-eared opossum, Brazil is my article. For an introduction, rabies is almost 100% fatal and is caused by the rabies virus RABB. In Brazil, successful vaccination has eliminated dog-associated rabies. Now, non-blood-feeding bats are the primary rabies reservoir in urban environments. White-eared opossums live in urban areas, but are considered resistant to rabies, with rare reports of low body temperature. The research questions that we have whether white-eared opossums can naturally become infected with rabies, which rabies virus lineage infected the opossum, how many rabies-positive bats and opossums are distributed in an urban environment, and whether ecological overlap between bats and opossums can explain viral spillover. The hypothesis in the study is the researchers hypothesized that spillover transmission from urban bat population can infect white-eared opossums despite their presumed resistance to rabies. Methods that this article used when studying rabies would be brain tissue from 930 bats and 22 opossums was tested by direct fluorescent antibody testing and confirmed by virus isolation and cell culture. 15 of the 22 opossums were also tested using RT-PCR and phylogenetic analysis of the Rabies or rabies virus glycoprotein gene to identify the lineage. Immunohistochemical analysis was done on the opossums to determine which organs contain rabies virus antigens. Brain tissue was used to look for neurological disease as well, or like any damage. Geographic maps, GIS, were made to show where rabies-positive bats and opossums were found, defined where they overlapped. And so spatial analysis is a great tool in rabies surveillance, by the way. Of the 22 opossums tested, only one opossum was positive. The female opossum had paralytic rabies and had neurologic signs before death. Histopathology revealed mononuclear meningoencephalitis. The rabies virus antigen was found in her cerebrum, cerebellum, heart, liver, and adrenal glands. Researchers sequenced the glycoprotein G gene of the rabies virus from the one infected white-eared opossum and compared it with rabies virus sequences from different animal species in Brazil. The phylogenetic tree groups rabies strains based on genetic similarity. Each color represented a rabies virus lineage associated with a different animal. The red label represents the virus from the white-eared opossum. The opossum virus clustered with the Artibius fruit bat lineage. Interpretation of the graphs that they had in the study. The virus infecting the opossum was genetically most similar to Artibius fruit bat rabies viruses. This means the infection most likely originated from fruit bats. It relates to the methods because the result came from the RT-PCR sequencing and phylogenetic analysis done on the rabbit opossum. 30 of the 930 bats tested were positive for rabies. That's 3.2%. 57% of them were Artibius, and 43% of them were insectivores. Researchers used geographic information system mapping and kernel density analysis to determine where rabid bats and opossums were located in Campinas, Brazil. The map shows green as low concentration, yellow as medium, and red as high concentration of Artibius bats. The black stars, the rabbit opossums, and the other symbols are concentrations of other bat types. Interpretation. The rabbit opossum was found inside an area with a high density of Artibius fruit bats. Fruit bats and insect-eating bats overlapped in many urban areas. The infected opossum lived in a vegetated area with a dense Artibius bat population. Method relation. The figure shows spatial analysis, which researchers used to compare locations of captured bats and opossums to identify where spillover transmission was likely to occur. The authors concluded that white-eared opossums are susceptible to naturally acquired rabies despite their resistant reputation. The phylogenetic and geographic analysis suggested that fruit bats are the source of infection and that rabies spillover can occur between wildlife species in highly urbanized environments. Rabies was observed in organs outside of the brain, suggesting systemic spread, which supports the clinical signs of paralytic rabies, which are opossum bats. Because opossums frequent areas near homes and often interact with domestic animals, the authors point out that spillover infections could make new opportunities for rabies to transmit to people and their pets. The study concluded that continuous wildlife surveillance, spatial mapping of rabies-positive animals, and a One Health approach are important for detecting emerging rabies transmission and preventing infections. The author suggests that white-eared opossums may acquire spillover from urban bat population. How might increasing urbanization and habitat overlap between wildlife species affect the emergence of new zoonotic diseases? What strategies could public health agencies use to reduce these risks? The study challenges the belief that opossums are resistant to rabies. Should public health surveillance programs change how they monitor species that are traditionally considered low risk for disease? What evidence would be needed before changing surveillance priorities?